Все?
Там вы готовы. Здесь вы запись начали. Здесь вы стартовали.
Good afternoon, dear students. Lecture number 4. The topic of the lecture, Ontogenesis. General patterns of human embryonic ontogenesis. Critical periods of ontogenesis. Plan of the lecture. At first, ontogenesis. Types and periods of ontogenesis. Secondly, embryonic period of ontogenesis. Fragmentation and blastula formation. Gastrulation. Differentiation of germ layers. And the next one, provisory organs. Features of human embryonic development. Regulation of ontogenesis. In conclusion, critical periods of ontogenesis. In the first half of the 20th century, a part of biology was formed. It called a living developmental biology. It studies the hereditary, molecular, and structural basis of development, as well as the mechanisms of regulation of ontogenetic change at all stages of the life cycle of an individual. The term ontogenesis was proposed in 1866 by Ernst Haeckel. So, individual development of organisms or ontogenesis is the sequential development of each individual which is based on the implementation of genetic information at all stages of existence in certain environmental conditions. There are two types of ontogenesis. Indirect, low-level occurs in insects, and direct non-low-level, typical for birds, and direct intrauterine, typical for mammals. Period of ontogenesis. At first, progenesis or pre-embryonic period, embryonic period, and post-embryonic period. The pre-embryonic period or progenesis includes gametogenesis and fertilization. Embryonic period includes fragmentation and blastula formation, gastrulation, histone and organogenesis. And post-embryonic period includes pre-reproductive, from birth to puberty, reproductive, puberty, and post-reproductive, decline of sexual functions, aging, and death. The pre-embryonic period of ontogenesis you study in detail on the practical lessons. The post-embryonic period of ontogenesis you will study during all your learning in the university, when we will study therapy, surgery, and other disciplines. Today, we will speak about the embryonic period of ontogenesis. So, embryonic period, fragmentation or cleavage. At the stage of fragmentation, mitotic division on the embryo cells occurs, in which the genetic information is exactly distributed without the growth of the daughter cells, followed by the DNA reduplication. There are practically no G1 and G2 periods in the interphase. Cells, blastomeres, decrease in size. The total volume of the embryo's body does not change, but it becomes multicellular. The biological and genetic meaning of fragmentation is that as a result of repeated cycles of reduplication and separation of DNA, the genotype multiplies. The human germ fragmentation begins to the end of the first day and continues during 3rd to 4th days after the fertilization, when the germ inside fallopian tube to uterus moves. The germ movement provides by the peristaltic muscles fallopian tube contraction, flickering cilia of his epithelium, and by moving the secretions of the fallopian tube glands. The germ nutrition provides by small reserves of yolk in the ovum and the contents of the fallopian tube. The human zygote fragmentation is full, uneven, and asynchronous. The first division ends in the 30 hours and 2 blastomeres form. After the stage of 2 blastomeres, the stage of the 3 blastomeres comes. In 40 hours, 4 cells form. By the first divisions, 2 types of blastomeres form, dark and light. The light blastomeres split more quickly and arranged in the single layer around the dark ones, which are located in the middle of the germ. From light blastomeres forms which connects germ with the maternal organism and provides the nutrition. The dark blastomeres form embryoblast. It forms the body of the germ and all other extraembryonic organs. By this time, the germ consists of 7 to 12 blastomeres. By the 4th day after fertilization, a cavity appears in the morula. It turns into a blastocyst. Within the 3 days, the blastocyst is located in the oviduct, fallopian tube. After 6 days, the blastocyst enters the uterus. By this time, it consists of 107 cells. Afterwards, the initial period of intrauterine development begins. How is it going? The blastocyst covers a fertilization shell. There are many lysosomes in the trophoblast cells. They destroy a fertilization shell. The penetration of the germ into the uterus is called the implantation. It begins by the 7th day after fertilization and continues during 40 hours. There are 2 stages of implantation. First, adhesion, sticking, and second, invasion, penetration. On the 3rd stage, the trophoblast linkages with the endometrium, mucous membrane. During the 2nd stage, the trophoblast destroys this membrane and trophoblast penetrates into the uterus, destroys its epithelium, the underlying connective tissue and vessels. The trophoblast comes into direct contact with the blood of the maternal vessels. The implantation fossa forms. In 2 weeks, the trophoblast uses the product of the decay of the maternal tissues, histiotropic type of nutrition. Afterwards, the germ nutrition provides direct from the maternal blood, gematotrophic type of nutrition. The period of implantation is the first critical period of germ development. The next stage of embryonic period of ontogenesis is gastrulation. This is a phase in which cell material is distributed into the 3 germ layers. Germ layers are dynamic clusters of cells which characterize a separate spatial arrangement. There are 4 mechanisms of gastrulation: invagination, epiboly, emigration, and delamination. There is a mixed type of gastrulation in the human body, so there is a combination of mechanisms described above. Phases of gastrulation. The first phase occurs by delamination, with the cells splitting into epiblast and hypoblast. On the 7th day, the extraembryonic mesenchyme begins to develop. The mesenchyme grows to the trophoblast and it's embedded in it, forming the chorion. Phase 1 ends with the formation of germ layers and extra germ membranes chorion and amnion. Стоп, погасло все. Нажмите на него. Ну это смешно просто. Ну просто смешно. Что случилось-то с ним?
Уснул?
Так, хорошо, ладно, я поняла. Амнион и корион. Ну, слушайте, ну это вообще сегодня конец. Так, давай дальше что мы?
С какой
Ну вот the second phase, да.
Угу.
Нет, чуть-чуть пониже. Я лучше... Нет, пониже текст. Угу.
Нога падает просто опять.
Ну, слушайте, это вообще прям ужасно, конечно.
Ужасно.
Это все, запись у меня здесь все идет. Вырежешь.
Ничего страшного, конечно, вырежу.
Так, чуть пониже текст. Ага. Чуть пониже. Не. Ага, да.
Начинаю, да?
Угу. The second phase of gastrulation continues to 17 days of development and ends of the formation of a complex of axial organs. There are neural tube, cord, and intestinal tube. This process provides by the regular distribution of cellular material. At this stage, specific proteins are synthesized and direct cell differentiation. In the period between the first and second phase of gastrulation, the extra embryonic organs active forms. The placenta and chorion form. This is called placentation, and this is the second critical period of germ development. The next stage of embryonic period of ontogenesis is organogenesis. This is a process of forming organ systems in embryonic development. It includes two phases. The first phase is neurulation. Neurulation is formation of the neural tube, cord, and intestinal tube. And the second phase is the phase of building other organs, the formation of internal and external organization features. The development of organs characterized progressive changing of their form, the composition of tissues and biochemical and physiological cells features. From the material of the external germ layer, the epidermis and its derivatives, components of the organs of vision, smell, hearing, and tooth enamel develops. The endoderm forms the epithelium of the stomach and intestines, the cells of the digestive glands. The cellular material of mesoderm is distributed among several cymaters. Dermatome, which forms the connective tissue of the dermis. Sclerotome, given the skeletal structure. Myotome, skeletal musculature. Nephrotome, rudiment of the urinary and reproductive system. The mesenchyme, ectoderm, and mesoderm forms all types of connective tissue, smooth muscles, blood, and lymphatic system. This classification is conditional because formation of each organ take part in several germ layers, with the advantage of one of them. Strictly deterministic structures develop from certain germ layers. The differentiation is the process of transformation the germ homogeneous cell material during embryonic ontogenesis into stable structures that are heterogeneous in their morphology, biochemical parameters, and functions. During the embryonic development, the connection of the embryo with the external environment is necessary. It is provided by special extra embryonic organs that functions temporarily. These organs are called temporary or provisional. These include the chorion, amnion, yolk sac, and allantois. In the human body, the yolk sac and allantois reduced. останавливает низкие уровни заряда. Ну, это просто, я не знаю, это вот все, что можно придумать, все сделается.
У вас ВВС запись еще идет?
Идет.
Угу. Не останавливайтесь.
Ну, да я знаю, что останавливается. Я не знаю, что еще нужно продумать, чтобы все было нормально.
Да, вроде смотришь, ведь со всех сторон.
Я не знаю, что еще надо. Ну, здесь вот еще надо, чтобы хуже было. Так, надо сейчас понять, с какого места это все выскочило. Немножко текста вниз прогони, пожалуйста.
Так.
Ну,
Еще?
давай вот it's provided by special extra. Вот после necessary. Да, вот здесь вот. Ага.
Угу.
Нет, ну это вообще сегодня просто.
Так. На текст смотрите, да?
Угу.
Ничего не мешает, не закрывает?
Нет, ну закрывает телефон верхний угол полностью. Меня это будет отвлекать. Еще лучше бы сдвинуть в сторону.
В сторону?
Ага, потом у меня глаза поедут, да.
Так, в сторону глаза поедешь. Сейчас я пониже его еще. Тихо.
Да, вот так нормально.
Не стоит. Врагу не пожелаешь, честное слово.
Да, вообще ужасно все сегодня.
У нас там половина лекции еще даже не записалась, да?
Ну, нет, мы движемся уже к концу.
Конечно, все прекрасно. Так. Есть?
Угу.
Нет. А там много еще?
Записывать?
Да.
Ну, есть.
Я его вставляю, боюсь, что вместе.
У нас ОБС-то не остановится.
Не остановится. ОБС точно не должен.
Так, интернет отвалился.
Здесь?
Оффлайн. Ну что? Ну, что можно сказать?
Так, подождите. Вот эта нога сейчас столько времени отнимает, вот пока я ее настрою, чтобы она встала как надо.
Нет, почему он тухнет так быстро, я не могу понять. Вот он зачем затух? Сейчас оффлайн пишет. Нет, ну Ваня, конечно, получит по голове опять очередной раз. За то, что просила по-человечески сделать. Так, вот сейчас хорошо.
Ага, сейчас закрывать будет вам?
Нет, нормально. Нормально.
Не закрывать, да?
Нет, абсолютно нормально.
Так.
Давай тогда я сначала, что ли, during the embryonic development.
Сейчас мы посмотрим, что там за оффлайн.
А, да.
Почему оффлайн? Нет, видите?
Угу.
Так, отсюда, да?
Да. Главное, чтобы только он не оторвал.
Нормально?
Да, вот с 20-го слайда получается.
Так. И, пожалуйста, розеточку прикройте. Да. Так, здесь. Все. Начинаем?
Угу. During the embryonic development, the connection of the embryo with the external environment is necessary. It is provided by special extra-embryonic organs that function temporarily. These organs are called temporary or provisional. These include the chorion, amnion, yolk sac, and allantois. In the human body, the yolk sac and allantois reduced. The yolk sac is a part of placenta, and the allantois is a part of umbilical cord. So let's speak about the amnion and chorion. The amnion. The amnion cells secrete the fluid in which the embryo develops. The amnion fluid is the protein, polysaccharides, and mineral salt solution. This is protect the embryo from mechanical damage. The chorion is external shell. It develops in 7th to 8th days of embryogenesis. This considers this the beginning of the implantation. On the chorion there are many vessels which penetrates into the uterus mucous shell. In the end of 3rd week, the blood vessels appear. The place of the greatest branching of the chorionic vessels and their closest contact with the uterus is called the placenta. In a human body is a hemachorial type of placenta. The germ body connects this placenta through the umbilical cord, which includes the blood vessels. There is a placental blood circulation. The embryo blood never mixed with the mother's blood. It washes the chorion vessels, but in normal situation never penetrates into the embryo capillaries. The supply of embryo of the nutrients, oxygen, elimination of waste products provides by the placenta. There is a specific cell barrier through which microorganisms and a number of substances from the mother's bloodstream are not passed. The damage of this barrier leads to fetal abnormalities and pregnancy pathology. The placenta is not barrier for drugs, alcohol, industrial and food poisons, foreign proteins, antibodies, and certain viruses. The process of human embryogenesis continues during about 265 to 280 days, or 38 to 40 weeks. So the human embryogenesis can be divided on the 3 periods: initial, pre-embryonic, 1st week; embryonic, 2 to 8 weeks; fetal, from the 9th week of the development to birth. By the end of the fetal period, the laying of the main embryonic rudiments of tissues and organs ends, and the embryo acquires the main fetus characteristics of a human. By the beginning of 9th week, length of the embryo is 40 mm, weight is about 5 grams. So, brief chronology of human embryonic development. During 3 days, moving through the oviduct with fragmentation. Day 4, blastocyst. Day 7, implantation into the uterine mucosa. Day 14, placenta. Day 16, the germ mesoderm is formed. At 3 weeks, the heart, blood vessels, and pronephros, the precursor of the kidney, are already laid. By the 4th week, the child has laid almost at vital organs, nerve ganglia, a prototype of the brain, lungs, stomach, liver, pancreas, endocrine glands. By this time it has formed a primary kidney, mesonephros, rudiments of hands and feet, as well as 4 pairs of gill arches. At the 5th week of gestation, the embryo forms a bladder, rectum, gonadal ridges, bronchi and lungs continue to develop, and the final kidney, metanephros, is laid. At the 6th to 8th week, the child's face is formed. It is already possible to see the emerging eyes and ears, as well as fingers and toes. At this time, there is an intensive growth of the head, which can be half of the entire length of the trunk. The size of the baby at the 8th week is about 3 centimeters. By the end of embryonic period, all major organ systems are differentiated. After that, the fetal period begins. During this period, the embryo length and weight increase and it acquires the main fetus characteristic of a man. At 24 to 25 weeks of gestation, the length of the fetus is 30 centimeters and the weight is about 700 grams. At 28 weeks, all the body's organs are already formed, but not quite mature. Its length is about 35 centimeters and its weight is about 1000 grams. During this period, the child has a chance to survive in case of a premature birth. The fetal period ends with the childbirth in the 38 to 40 weeks. Regulation of ontogenesis. There are the following levels of regulation of ontogenesis: gene, epigenetic, cellular, histic and organ, organismic, and biocyanotic. Gene level of regulation of ontogenesis. The genes that control the cause of ontogenesis are chronogenes. This is the genes which control the time of the occurrence of events. They form during the amplification process in the ovum. The amplification is a process of increasing the number of genes copies. Amplified copies begin to be translated immediately after fertilization and act at the earliest stage of development. The embryo's ovum genome is inactive until stage 2 to 8 of blastomeres, and the composition of all the resulting proteins is controlled by the mRNAs stored in the ovum during its formation. The composition of these proteins depends only on the mother's genotype. Activation of the mammalian embryo genome begins with the 4 blastomeres. The second group of the genes, homeotic genes. They determine the process of the growth and differentiation. This is a terminal differentiation genes. They provide the cell differentiation. And the third group is segment polarity genes. Control the left-right symmetry, determination of the polarity of the central nervous system, the formation of the skeleton. Usually the mutation of these genes are lethal. There are 4 genes, mutation of which are associated with impaired formation of the brain and spinal cord. They are on a screen. So, the functions of genes are different. Some of them encode the protein biosynthesis, tRNA and rRNA synthesis. And another part of genes control the processes of differentiation, determination, and morphogenesis. The next level of regulation of ontogenesis is the epigenetic level. The epigenetic regulation is a process that changes the activity of a gene without changing its coding sequence. The epigenetic gene's modification is one of the mechanisms which provide differential gene expression of different cells and tissues during the ontogenesis. The main role in epigenetic regulation is played by DNA methylation, histone acetylation, synthesis of alternative RNAs or antisense transcripts. DNA methylation is the process of connection a methyl group to a cytosine. The methylation causes a transcription block. The active gene associated with hypomethylation or the lack of the methylation. The non-active gene associated with hypermethylation. Acetylation of the histones is the process of connection a acetyl group of the histones to the histones. Positively charged ends of histone proteins interact with negatively charged DNA phosphates. Acetylation weakens interaction with DNA and allows binding to certain transcription factors. And synthesis of alternative RNAs. A significant proportion of non-expressed genes, up to 15%, associated with intense transcripts, antisense transcripts. These antisense RNAs do not perform coding functions. Cellular level of ontogenesis regulation. The gene level of ontogenesis regulation is realized through cellular processes. Proliferation, differentiation, moving, sort, selective cell death, and cell adhesion. Histonic and organ levels of ontogenesis regulation is realized through the embryonic induction. The embryonic induction is the contact between the inducing and responding cells. During this process provides diffusion of the substances from one cell to another, and the matrix of the one cell induced changes in another cell. Organismic level of ontogenesis regulation. Neurohumoral regulation. It is carried out due to the joint work of the nervous, endocrine, and immune systems. Regulates the growth, development of the body, its sexual differentiation, and reproductive function. Biocyanotic level of regulation of ontogenesis. It is carried out with the help of environmental factors. Mutagens are factors that cause changes in the hereditary material of cells, mutations. And the next one, teratogens, are factors that affect the embryo and fetus and cause malformations and abnormalities in postnatal development. Well, let's watch the video. Fertilization occurs in the ampulla part of the fallopian tube. To merge with the ovum, the spermatozoon must overcome two membranes, the radiant crown and the zona pellucida. To overcome the zona pellucida, the spermatozoon uses acrosomes enzymes. An acrosome is a modified lysosome. After the first spermatozoon has penetrated, the rest number of them are not able to fertilize the ovum. After the penetration of the spermatozoon into the ovum, a zygote is formed. After that, the zygote begins to form nuclei. Each of the pronuclei of zygote, male and female, has a haploid set of chromosomes. The paternal chromosomes are located in the male pronucleus, and the maternal chromosomes are located in the female pronucleus. Having formed at different ends of the zygote, pronuclei move towards each other, after which the membranes dissolve and the chromosomes line up in the metaphase plate of the first mitotic division. The fertilized ovum moves inside the fallopian tube to the uterus. We see the fragmentation process. Now the trophoblast is in the uterus and the implantation fossa forms. This is a stage of implantation of germ in the uterus. For better contact with the endometrium, the blastocyst frees up from the zona pellucida. After that, the cells of the trophoblast, the surface layer of the blastocyst, eject finger-like outgrowths for immersion in the endometrium. The stage of gastrulation begins and distribution of cells material occurs. Now we see the organogenesis. The neural tube, cord, and intestinal tube formed. We see the heart beating. The arms and legs are formed. The orbits move to the front of the face. And the embryo acquires the main features characteristics of the human. Now this is the embryo brain. There are many active processes in it. The associative linkages between neurons form. The child can see, hear, feel during he or she is in mother's body. And now we see another period of ontogenesis, the birth. I hope this child is healthy and lucky.
Скажите мне в голос просто спокойно.
Все, дальше мотаем. Дальше. Вот, все, поехали. And the next point of the lecture, critical periods of ontogenesis. There are the critical periods of ontogenesis in which the organism is most sensitive for negative factors. Each stage of ontogenesis begins of period of qualitative restructuring, which occurs by changes of the determination, proliferation, and differentiation processes. In these periods, reduced resilience to mutagens and teratogens provides. There are several critical periods of human ontogenesis: gametogenesis, fertilization, implantation, 7 to 8 days of embryogenesis, formation of a complex of axial organs and placentation, 3 to 8 weeks of development, stages of reinforced growth of brain, 15-20 weeks, formation of the main functional systems of the body and differentiation of the sexual apparatus, 20th to 24th weeks. And the next one, birth, neonatal period, first month of the life, puberty, 11 to 16 years, menopause in woman. As we see, the most of critical periods of ontogenesis are in the embryogenesis. We know that out of 300 million spermatozoa, ovum is reached by several thousands. And of the 100 zygotes formed, 95 died during embryonic development. But early infant mortality is only 2 to 3%. As a result of embryogenesis violation, the malformation and abnormalities form. According to World Health Organization, the abnormalities of development occur in 1 to 2% of people. Thank you for your attention. Слышно? Сейчас я выключу. Я не понимаю, студенты решили